share my video on phenomenalstemist. Just give me a second. Hey, everybody, how are you? Just come in the room. Take your time to come into the room. Good afternoon. Akuna, how are you?
I am well. How are you doing?
Yeah, I'm doing good. I'm doing good. I'm just glad that you have agreed to do this interview. We're really going to have a great time. Hey, everybody. This is Dr. Akuna Ahanacho. She is a scientific director in the biotechnology industry. Awesome. Listen, this is a Black woman right here. Scientific. She's going to talk about what she does in the future. But right now, I'm just going to give a brief bio and just learn a little bit about you, Akuna. Number one, Dr. Hanacho received her PhD in cellular and molecular physiology from Johns Hopkins University School of Medicine. Woo! I got a couple of you watching. Listen, I need some likes and some loves. I need you to share this. Not only did she receive her PhD, she also received her master's degree in chemistry from the University of Virginia, and of course,
Of course, of
a
course.
bachelor's in science in biology, just like me, same year, from Oakwood College.
Yes.
Oh, see.
OC, OC.
Now University in Huntsville, Alabama. Akuna, thank you so much for joining us on Phenomenal STEMist.
Oh,
We're
you're
going to get
welcome.
right into it because I know we have a lot to talk about. We're going
All
to talk
right.
about how your role as scientific director in the biotech industry, your process to becoming that, and just we're going to dabble a little bit in understanding the processes of vaccine development, seeing, this is a hot topic right now. So before we go into that, number one, my first question to you is, when did you fall in love with your field of study?
Oh, man. I have long had a love affair with science and learning in general. So, as a child, even, I was conducting these rudimentary science experiments in my backyard. And I was always taking things apart and trying to put them back together so that I could figure out how they worked. So my love affair with STEM and with science and engineering, technology, it goes way back.
Way back,
Yeah.
way back, just like me, backyard. I was dissecting flowers. I was looking at the carpel and the stick. I was doing all that
Yes.
kind of stuff.
Yes.
I tell parents all the time that it starts from young. It starts from
Hmm.
young, and I think what it did was it removed the fear. We weren't
Yes.
afraid of it.
Yes. And my
Yeah.
parents encouraged it. So they weren't
Mhm.
fussing, oh, look what you did to the flower garden. You tore up the patch, or look what you did over here. We had a vegetable garden back there as well. So I was always walking behind my dad, seeing how he planted the seeds and how things were growing. And they encouraged that
Yeah.
wonder, that curiosity. So that definitely aided in my development and my desire to just try to learn more, because they didn't stifle any of that.
Yeah. You hear that, parents? Very important. Right, especially when the boys break something and try to put it back together. That's an engineer
Right in
in
the
the
making.
making. That is an
Yes.
engineer in the making. So what were your struggles in school? So, I know you went to Oakwood just like me. So you could
Hmm.
talk about your struggles in college. You could also talk about your struggles in graduate school. What do you think your struggles were, and how did you overcome those struggles?
So my struggles, I would say my struggles started in graduate school. It was definitely completely different learning environments. I went from a historically Black college in both instances to universities that were not, where there wasn't a great representation of minorities. We were
Right.
definitely underrepresented in across all departments. So not even specific to the chemistry department at UVA or the physiology department at Hopkins, or the School of Medicine, for that fact, at Hopkins. So one of the most challenging things for me, in graduate school, actually, was the inherent uncertainty that is kind of baked in or built into obtaining a graduate degree. No one had sat me down and talked to me about that. And so, one, coming to that realization that there's a lot of uncertainty. There are parameters, but there are there are boundaries that are kind of wide open. When you're working on your master's thesis or when you're working on your doctorate, it's pretty much open. You're trying to figure out something that's novel and new, and it was different not having confined parameters within which to work.
Yeah.
So another thing that came as a shock to me was the realization that 99, and yes, I did say 99% of the time, your experiments are not going to work.
Yeah.
They're just,
I'll tell you.
they're not going to work.
Yeah.
So coming to that realization and figuring out that yes, your hypothesis was disproved. Now pick yourself up and figure out what your next what
Dry
your
your
next step is
tears.
gonna. Right, right. So
Toya is
but
watching. Toya is watching. Hey, Toya. She said,
hey,
and of course. Oh, see. Go ahead. I'm
oh,
sorry.
definitely. So those came as great shocks to me, and
Yeah.
now when I do STEM outreach, I try to make people aware of those potential challenges. And I mean, the circumstances might be different for them and the path that they choose, but um, I think having that foreknowledge, it just makes you better prepared. So, in my graduate program, there were different biochemical techniques that I was using. So, for my doctorate, I used a combination of mouse models, structural biology, and other biochemical or biophysical techniques to
Mm.
answer these, scientific questions. So, for structural biology, it focuses on, or it uses as a tool, crystallography. So, for those to actually perform crystallography and start to look at different chemical interactions that are occurring on the molecular level, I first had to set up crystallization screens. And I
Mm.
set up countless crystallization, I mean, more than hundreds, probably thousands of different crystallization screens in which I didn't get a single crystal.
Oh, it's painful. I don't think you guys understand how hard the process of X-ray crystallography is just to get a protein to not denature. I know I'm
Right.
using big words, but listen, guys. Imagine you spending a lot of time on something, hours on something, hoping that you'll get a result, and you don't get it. And your PhD or your master's is dependent upon it. Listen
Yes.
what Akuna is saying. It's not like college. You do all your work, you make
Right.
all the good grades. It's not even like medical school where you pass all your tests. You can do all the good work and pass all the tests, but if random studies don't work, you
Yes.
can be there 4, 5, 6,
Ever.
8,
Forever.
9 years.
trying trying to get, because at the end of it, you want data that's going to tell a story.
Right.
Um, and without sufficient data, you can't tell that story. So they will keep you there until you have
Until
enough data to tell a story. So,
mercy.
and I'm talking about setting up the crystallization screens. Before we even get to the point where I could set up the crystallization screens, I first had to purify protein in sufficient quantities to
Yeah.
even set those up. So, I mean, there were steps with that because I'm trying to figure out what's the best way for me to produce this particular protein. What host system am I going to use to produce it? What species am I going to use to produce it in? Is it going to have all of the different post-translational modifications if I'm not using bacteria, if I'm using some other eukaryotic system to try to produce these essentially human proteins? So it was a challenge. It was a struggle
Mm-hmm.
figuring out the production, the expression, and then once getting to the point where you purified in sufficient quantities to even set up the trays, realizing that sometimes those screens didn't work, and
Oh,
going back to the drawing board and figuring out, okay, what's next? Or with my mouse experiments, so I had mice in which I introduced a mutation. And so even just introducing that mutation, developing a cohort, setting up different breeding cages so that I could, breed the mutation into these mice. There were times in which I would challenge them with different things, and the data, there was no clear effect. But that
nothing.
was not months. I mean, like literally years of work getting to that point, just for at the end of it to say, well, there seems to be this trend that we can observe here in this cohort of like. So I mean, it was definitely a challenge and a struggle trying to translate those discoveries and trying to be excited about those accomplishments and see how things fit into the larger picture. But these were not discussions that anyone talked to me
Told
about
you.
before. Nobody told me this.
Yeah.
But yeah, science can be a cruel master. It can be a cruel
Listen,
master.
I call it a boring. And I'm not trying to say we shouldn't become like a phenomenal STEMist, right? But
Yeah,
I want us to be honest because we're talking about the struggles.
yeah,
I tell my students all the time it's a boring roller coaster.
yeah.
Graduate school to me was a boring roller coaster, meaning it was lots of lows, and then you have this high
Yes,
point, and then it
yes,
drops down,
yes,
and then you coast for another couple of months, and then you get a
yes,
result,
yes,
and then you don't get a result,
yes,
and you have to learn to manage the ups and downs. You
yes,
have to
yes,
learn to manage it.
yes. Managing your expectations and then
Yeah,
also rewarding yourself or celebrating your triumphs, however
yeah.
small they might be.
However
Um,
small and fast. However
yes,
small and fast, and that's a very important life lesson. That is a very important life lesson that in life you're not going to always get what you want. So,
yeah.
especially when I tell my students who are in lab and they're, oops, I lost you. I lost her. Hopefully she'll come back. So anyway, yeah, got you. You're back. You're back. You're
Okay,
back. Yeah.
okay.
In lab where they expect a certain result and like, oh, it didn't work. I'm like, well, welcome to science. Welcome to science. Yeah. Okay. So basically, your deeper struggles was the uncertainty you had
Yeah.
because like you were doing well in school,
Oh yeah.
in Oakwood, and you were getting the good grades, and now it doesn't matter how great your grades are.
Right.
Like this
Yeah, my
is
grades
this
didn't
is life.
matter. The lab didn't care. Like my mice didn't care what my class scores were.
Right.
My crystals didn't care. My crystallization screens cared not about how much effort it took for me to get to that. No, it didn't
No,
care.
right.
It didn't. It didn't care at all.
Yeah.
Another thing that was challenging for me in graduate school was how isolating just the whole graduate experience could be at times as well. Um, the work I was doing with structural biology, it was it's very different from some other fields of study. So even just trying to have a conversation about some of the challenges that I was having would sometimes prove difficult if I was talking to someone who wasn't necessarily in the same department or had an understanding of what it took. And then outside of that, like my family and my friends who weren't in science at all, they had no particular understanding of the challenges that I faced. So they would try to be encouraging, but
They should listen.
um,
I
yeah,
would tell my church issues. Like my church knew about bacteria not
yes,
working. Like
yes. No, really, my community of faith was so important. My church folks they helped to keep me grounded. So family, friends, community of faith, they all helped to keep me grounded. So yeah, I tried to explain. And another thing too that helped strengthen my communication skills, because I mean, you can't go in and talk all this big, words that you typically use. You have to explain it in terms that everybody could understand. Um,
yeah,
yeah, yeah.
yeah,
Another thing too that came out of, I guess, a larger group of us that were starting to feel isolated and disconnected. We came together and we formed an organization called the Biomedical Scholars Association. And essentially it was just a group that consisted of Black and brown graduate students who we were from different programs. We weren't all in the School of Medicine. We were in different programs at different stages of matriculation. And we started sharing our experiences as a community. And we
yes,
began to support each other. And we came together and we said, okay, these are some tips. So the next time you have your thesis committee meeting, you should maybe think about these things. Or it might be good for you to meet with your thesis committee members before.
yeah,
Talk to them before you have your regularly scheduled meeting just to see if they have any input or any suggestions. And then we would also have these impromptu discussions in which we just needed to vent. Like if we just needed a vent session about what was going on personally, professionally, whatever was going on, we would come together. And then out of that, we grew this network of people where we started to have committees that would come together and help you get your study preparation, or as you were
yes.
preparing for your mock oral
Mock.
examinations.
Yep.
We would come together and we'd give test questions and we'd try to challenge them and have them think about things in different ways so that they could be better prepared. And essentially we developed the network that we needed. So, and sometimes there were advisors that helped us who weren't in our department, didn't have anything to do with our work, but that just took a personal interest in us.
Yeah.
Um, And as they took a personal interest in this, they would give us tips about how to navigate through the space at Hopkins or how to network professionally. Because a lot of the people that I met there, I still reach out to them. They're the
Right.
people who will serve as panelists for some other workshops that I'm doing. There are people that helped write recommendations for me later. There are people that helped to establish my diverse professional network so that across different fields, we can all communicate and we can talk and we can keep paying it forward. So that was another skill that came out of that, just learning how to network and come together. And if you're not getting what you need, building what you
Build
need.
it. That's right. What you're saying is so. I had very a similar experience. It was already built for me. So what you created, people later on are experiencing, Akuna, because we had, when I was there, it was called the Percy Julian Association. Percy Julian
Okay.
was a black biochemist, and
Mhm.
they named the people who started PJA, named it
Yeah.
after him. So when I came in, I just seamlessly came into that network. So it's so important that either you build that network for yourselves or
Mhm.
for people that come behind you. Because
Oh,
what you're saying
definitely.
is,
So
what you
now,
said is
and
so
now
true.
they have, the organization still persists till this day. So I'm
Exactly.
happy about
Of course
that.
it was. Yeah.
And then also they now have the Office of Diversity and Inclusion, which did not exist when I was there. So
Mm-hmm.
now they have not only the organization that started when I was there, but now they have these other resources that are available to them, because now it has become um, an important thing at the institutional level where they say, okay, for
Right.
retention, for support, we need to have these kinds of things available for all of our students. So yeah, it's good.
Awesome. Awesome. Well, I love how you had the struggles and you dealt with the struggles. That's what's going to happen in order to persevere, especially in graduate school where you're trying to get a PhD.
Mhm.
You have to know that they exist,
Mhm.
be ready for them, and then persevere through them. All right. So tell me a little bit of what you do now, right?
Okay.
You're a scientific director
Yes.
at a biotech firm, or like a CRO is what we call it. You can explain what that is
Okay.
and just explain a little bit what you've learned.
Sure. So, a contract research organization is essentially an organization that can provide a variety of different services depending on what the organization specializes in or the field that they specialize in. That can be a variety of different things. So, in my particular organization, we focus on viral safety and viral clearance. So just trying to handle all of the viral safety testing, novel assay development, virus purification efforts, or so for every pharmaceutical that has to undergo approval by the FDA, if it's made using, if it's a biological product that is made using, say, a mammalian cell line, it has to undergo viral clearance testing. And essentially, what that viral clearance testing does, you're trying to establish the safety and effectiveness of that particular therapeutic. So that's essentially the area or the niche that we specialize in: viral safety and viral clearance studies. So essentially, what you're trying to do, there's, whenever a new product is being manufactured, there are several different points at which viruses could be introduced or other advantageous agents could be introduced
Nice.
into it. And so what our testing does, we evaluate different process steps, and we want to make sure that we're clearing viruses from the process. So by the time that it gets to the point where it's going to be injected or it's going to be delivered to a human being, we know
There's
that it's
no
safe.
viruses in them and they're safe.
Right.
Girl, you're working with viruses. I'm just playing. I'm just playing. Wow, Akuna, this is awesome. So you like, what have you learned in your process of being like, you're a scientific director? Like, what is,
Well,
talk about
even
that?
that was a jump, right? So I guess let me back up a little bit and talk about once I left, once I had my doctorate in hand and I left Hopkins. So I went to another contract research, and that was actually a contract research and development organization. And at the time, it was called Paragon. It is since now called Catalent Gene Therapy. And essentially, what I did there, I developed gene therapies and I developed vaccines. That was the focus that of my work while I was there. And essentially, developing a manufacturing process to produce these different therapeutics. That's what I was tasked with. So sometimes it was a vaccine, sometimes it was an AAV gene therapy. There are a variety of different projects that I was able to work on and things I was able to kind of have my hands in. So I do have expertise in the development of large-scale manufacture of vaccines, gene therapies, and other biopharmaceutical products. And that's what I did
Go
prior
on,
to
girl.
joining.
Go on, girl. Sorry.
That's what I did prior to joining my current organization. So I really feel, honestly, that God really just opened up doors for me.
Wow. Yeah.
Because if you had told me back when I was still a biology major at Oakwood what I was going to be doing, I probably would have laughed at you like, no, I'm not going to be doing that. So when I just look at the path from master's to doctorate to from one CRO to another CRO in the biotech industry, the breadth and depth of my experience is large just because of my exposure to so many different things. Because my degree is not in virology, but that is really where my focus is now. It's focused on virology. My background is in physiology and looking at things on the cellular and molecular level. So looking at things down to the level of molecules and cells. And I think that framework is kind of the foundation upon which all of my
Anything.
understanding and knowledge is built on. So like you're saying, so I was able to take the lessons learned and kind of those critical thinking skills and that just add them to my arsenal or add them to my toolkit of skills. So it allowed me to understand a variety of different processes and kind of understand things from a big picture view as well as down to the level of molecules and cells as well.
But the thing is, like, not only so you've gotten yourself given yourself because you majored in biology and bachelor's, then you majored in chemistry for your master's, and then you got your PhD. You've given yourself, like you said, an arsenal, a wealth of information that put you could put anything on the foundation.
Yes.
Sounds like to me that you gave yourself a strong foundation, right? But now,
Yes.
but you also learned to build networks.
Yes.
Because
Yes.
it seems like with your job now, with your career now as a scientific director, you're not really doing hardcore science anymore, are you? Like, what are you doing now?
No,
You're
it's more.
the Go ahead.
Well, it's a mixture. So a lot of it is translational research, you could say, and a lot of it is, and even that is interesting because with both of the organizations, I was recruited to both of those organizations. So I never essentially did a job search where I was plugging in and saying, okay, let me look at the job description and see if this is what I want to do. I was recruited to for my first job out of graduate school before I had finished. So that was based on some contacts and some networks that I had. So a friend of mine was already working there, and he was like, "I know you're finishing up. Would you have any interest in this position? Could you meet with the hiring manager?" And I had already knew at that time, as I was wrapping up, that I did not want to do or pursue a tenure track position. So for me, in terms of securing funding and trying to make sure that all of my laboratory staff, I would be able to support them through grants, it was a hard time. That was during the recession was still in place, so it was hard for more senior level tenured professors to receive funding from the NIH. So I figured me coming out new, fresh, if they're having difficulties, it's going to be tricky for me. So I was even at that point, I had already made the decision that I didn't want to pursue a tenure track position. So this opportunity presented itself as I researched the company and looked at what they were doing and what the opportunities that would be available to me. I was like, oh, wow, this is exciting. Yes, this is something I want to pursue. And then with my the organization that I'm working with now, I was actually doing some work there for another company. And while I was there doing work for another company, they were like, well, we would like to chat with you about some opportunities that we have available here. And at the time, I kind of poo-pooed them off and was like, I'm not really interested. I'm happy where I am. I'm not. But then over time, as God would have it, again, that was just another door that he opened up for me.
Wow.
And it was another opportunity for me to, again, expand my skill set. Because at first, I was hesitant. I was hesitant because I said, well, I don't have a degree in virology. All of this work is focusing on virology and viral
Viruses.
safety. All of my background is in structural biology. My background is in molecular endocrinology. It's in physiology. It's not necessarily, all of the things I'd done prior to this were kind of adjacent to virology. And although I had some experience with immunology and, thinking about that aspect in terms of, like disease pathology, um, I just wasn't sure if this was going to be a good
For
fit for
you.
me. But then
Yeah.
as I thought more about it, I was like, but it'll be an exciting time for me to learn something new and to continue to
Mm.
directly apply the background that I already have. I said, okay, I've done some vaccine development now, so I've seen that side of it, and I know, about viruses in that regard and how we're trying to, um, why we develop vaccines. The whole point of developing a vaccine is you're trying to prime your body so that it can resist this potential threat. So I
Mhm.
said, well, why not? Let's take a chance and let's see how things shake out. And it's been exciting
It's been
and
great.
very rewarding. And then
Wow.
also the impact to public health. That's another thing why I decided not to pursue tenure track. I wanted to
Wait, just to give everybody
sure.
understand what tenure track is. That's basically, for instance, for me, I'm a tenured professor. That's basically she didn't want to go into academia, at academic science, where she would
Yeah.
become a professor at a university where you have to have your own grants to run a lab where people work under you. As she said, it's still a tough time. So it was a tough
Yeah.
time back then. It's still a tough time, and so she wanted to basically just be in the scientific industry rather than academic science. So just giving that's what tenure track means, on your way to tenure. Yeah, go ahead, Akuna.
And I was a little impatient too,
Yeah.
because I wanted to see the direct application of, okay, this is the research, this is what we have done. What is the direct impact to public health?
Mm,
And so with the first position at that first organization, I had a chance to see the direct application. I'm
right.
preparing materials that are going to be used for clinical trials. I'm preparing materials that will one day wind up being a pharmaceutical that gets injected or that gets ingested by, a human being, and that has a great potential to impact their health for good. So that
Right.
was another thing that was very appealing to me, the ability to impact public health.
Wow. Wow. Okay. So this is a great movement or progression into my last question, which is this process of vaccine development. Can you discuss that? Like,
Yes.
can you explain the process of vaccine development? Of course, COVID is freaking a lot of people out, and they're not sure if they should get a vaccine. Or I know for those of you, you could watch my former video talking about the different types of vaccines, but we actually have a professional, somebody who I went to school with, who knows the details of vaccine development. Could you just talk about that?
So there are a variety of different ways in which vaccines can be made. And I think you touched on this actually in the video that you referenced. So I'll just briefly touch on them again. So you can weaken the live virus. So for instance, measles, mumps, rubella, chickenpox, all of those vaccines were developed by weakening the live virus. You can inactivate the virus. You can use some chemical means to inactivate the virus and essentially rendering it ineffective. And the polio virus, hepatitis A, rabies, those are all examples of vaccines that were developed using that method. You could use a part of the virus. Shingles, hepatitis B, and the human papillomavirus, those were all developed by using a particular part of the virus. You could use a part of the bacteria. So, for instance, in the case of vaccines against tetanus
Tetanus.
or pertussis or diphtheria, they used a particular part of the bacteria. Or another way that you could do it is using the genetic code for a part of the virus. So, in terms of using the genetic code, so there are a variety of different viruses, and they can have different genetic content. It could be made out of DNA, it could be made out of RNA, and so you can either develop a vaccine using DNA, develop a vaccine using RNA, or you can use a viral vector um, as a vehicle. So the COVID-19 vaccines that have been approved to this point or thus far are all mRNA vaccines. And essentially, how they work is they are packaged into lipid nanoparticles, and the lipids themselves, they serve as a delivery system by using the lipid bilayer to get into the cells. And these mRNA vaccines, they were also engineered to be more stable. So typically, mRNAs are produced by your body every day. Your body produces them. And typically, when your body produces them, they are degraded within minutes
Quickly.
or, yeah, within minutes or hours. That's usually, but these particular mRNA vaccines, they were engineered so that they would persist for days as opposed to minutes or hours, that time scale. So now they're going to persist in your system for days. But then after that point, they will be degraded by your body. And the part of the virus that they are using is the spike protein. So why this is important, the spike protein is the protein that's on the surface of the virus. And the spike protein, in essence, it helps the virus attach to the surface of your cells. However, once your body produces antibodies against the spike proteins, they can bind to the surface of the virus and essentially neutralize it. And maybe I should pause or back up here to talk a little bit about immune response or immune memory. So memory B cells, they're responsible for making antibodies. Those are the cells that actually make the antibodies. And the antibodies, once they are made, they can actually circulate in your blood, and they'll essentially stop the virus outside of the cell. Whereas T cells, they are also another component of your immune system, and they're specialized to deal with infected cells. So think of it as antibodies, they're trying to help stop the infection outside of the cell. So before it actually gets into the cell and is able to do anything, antibodies want to neutralize it and neutralize that potential threat. Whereas T cells, they're specialized to actually attack or deal with infected cells.
Yeah, and so they basically call T helper cells will call other white blood cells to kill the infected cells, or they'll kill it themselves. And that response will a lot of the times, unfortunately, hurt us.
Yeah.
With the real virus. So the purpose of the vaccine is to basically attack the spike protein.
Yes.
Not the whole virus, just the spike protein, or remember, like you said, memory, the spike protein, so that when the real virus comes,
it's almost like think of it like a primer. So you're trying to train or teach your body before it actually has to deal with the actual threat. Yeah. So before you're encountering the virus, by telling your immune system to make these proteins, you're actually telling it, hey, make these and recognize this. Recognize that it's a potential threat, and you get a chance to kind of prime your system so that it's aware. Because what usually happens is how viruses are able to circumvent your system, your body doesn't recognize it as a threat. It doesn't recognize it as something other.
Until it's too late.
Until it's too late. So then it's already hijacked your system. It's already gotten into your cells, and it's started to replicate. Viruses are always looking for a host so that they can replicate and take over. That's how they were designed. That's what they do. So by priming your system, you're helping your body to be aware of the threat, and it's almost like you're teaching it to, you need to recognize this and you need to neutralize it. So before this becomes a problem, here, this is what it looks like. Recognize it, neutralize it, so that it's not a problem for you later.
So do you trust, you've worked in the vaccine development process. Do you trust the vaccine development process? Do you, like, as a scientist? So many people are afraid. They think that you guys take shortcuts and what do you think as somebody who's actually in the field?
I trust the process. The FDA is very rigorous. They don't take shortcuts, regardless of whether it's a crisis or not. They don't take shortcuts. Neither do the other regulatory authorities in Europe and in other countries. So when you think about it, vaccines are usually made by first showing that they're safe and effective in experimental animals. So before it ever is part of testing in humans, you first have to show that it's safe and effective in experimental animals. And then once you establish that, then the vaccine becomes an investigational new drug.
And so it wouldn't even have made it to humans if a mouse or a primate, like a monkey, hadn't been tested on first.
Correct. This is just part of the process. And the process, I will admit, it was fast tracked for this virus. And there were a couple of things that fell in place. But to back up a little bit, let me talk just a little bit about the different phases. And then we can talk about how things were probably potentially fast tracked in this particular circumstance, as you know, in terms of the vaccine against COVID-19 or SARS-CoV-2. So in essence, once the vaccine has been tested in experimental animals and it's been established by the FDA or whatever regulatory authority is actually reviewing the data, and it's been determined that it's safe and it's effective, it will get this status as an investigational new drug. And then they will start conducting phase 1, phase 2, phase 3 clinical trials to actually see what is the long-term safety and what is the long-term effectiveness. So in phase 1, essentially, you test the vaccine on a small number of healthy adults. And you usually will have some placebo-controlled studies, and these studies are usually with small numbers. So let's say less than 100 people. And you're trying to answer a variety of questions. But the two main questions you're trying to answer are, does the vaccine actually generate an expected immune response? And is the vaccine safe? Those are the two main questions. So you'll have a small group of humans, less than 100 people, and you have a placebo-controlled group that doesn't actually get the vaccine, and then you'll have your other group that is getting the actual vaccine. And so that's phase 1. So during the phase 1 trials, the scientists at the pharmaceutical company, they have to study how to make the vaccine in a quantity large enough for preliminary trials. So while they're testing it in humans, they're also trying to see how can we produce this in sufficient quantities that we could generate enough for the population that we're trying to serve.
Which is a lot of people. This is the whole world.
Yes. And then another thing that you're usually trying to do in phase 1, you're also trying to establish, are there any adjuvants necessary to essentially boost or make you have or generate a stronger immune response. And essentially adjuvants are essentially any preservatives, stabilizers. Actually, no. Adjuvants they're given to elicit an immune response. So with an adjuvant, you know that you're going to actually elicit an immune response. So if you're adding that to the vaccine, you're doing that to sort of boost up or amp up the immune response that you'll get. And so usually during phase 1, they're looking at that. Are there any adjuvants that need to be added, or how are the adjuvants working? Are there any stabilizers that need to be added, or are there any other preservatives that need to be added to the vaccine? That's what's usually done in phase 1. And then moving on then to phase 2, they'll broaden the study. So this phase of the study, there'll probably be several hundred people that they're testing it. And they're going to still have those groups where you have a placebo-controlled group, and then you have a group that's actually getting the virus. And so during this phase,
vaccine.
Yes, sorry. I keep saying that. So you're going to have the group that's going to get
I did that last time too. It's common. It's common,
right?
Yeah.
You have the group that's getting the placebo, and you have the group that's getting the vaccine. I'll try to keep that straight. Don't want to confuse anybody. So during this phase, scientists are trying to determine the proper dose of the vaccine to be given. And they continue to study the vaccine's safety. So they also want to better define methods for manufacturing the vaccine, for stabilizing the product, and for determining the packaging. How is it going to be packaged? Is this going to be in vials? How is it going to be administered? And they're trying to establish the assays necessary for the trials. Because, at the end of it, when you're looking at your outcomes, you want to be able to see what are the actual impacts and what are the actual effects. So in phase 2, you're trying to sort out and figure out all of those things. It's a very important aspect to establish manufacturing consistency. You want to know that each batch, each lot of this vaccine is going to be consistent, so that with each, you have different release tests that you'll probably establish, assay release or lot release testing that you'll do to make sure that with each batch that's produced, you're expecting it to be safe and efficacious, and you're expecting it to have certain characteristics. Each lot should have certain characteristics. And so typically this can take 2 years, and sometimes it can take even longer. what is one of the biggest hurdles, I will say, in vaccine development is the actual production of the vaccine in sufficient quantities. Again,
Numbers.
going back to the population that you're trying to reach and how many doses that you're trying to produce or that you need to produce. This is the time when you're trying to work all of that out. So a lot of companies, right? It takes, so for instance, let's make this more real. So for the Pfizer vaccine, it costs them about $2 billion to develop this vaccine. And yes, I said billion with a B. It cost them about $2 billion to develop this vaccine. And they proceeded at risk. So what most companies do, they will make certain investments along the way, right? Because they want to make sure that they're getting a good return on their investment.
Yeah, yeah.
Right? So usually people are not going to front you. People are not going to throw a lot of money at the problem. With a lot of things in science, if you have enough money to throw at it, that can fix a lot of things. But a lot of companies, they're looking at their profit margins too. So they do want to make good impacts, but at the end of the day, they're like, we have to do this in a way where we're still right. Is this still going to be worth it for us to pursue this? So with Pfizer and BioNTech, they didn't accept any funding, any government funding. They proceeded at risk and they funded this completely themselves, the $2 billion completely themselves.
Did not know that.
One of the things that was going on in the background in their pipeline, Pfizer had already entered into a collaboration or a partnership with BioNTech, in which they were trying to develop a flu vaccine. And they were using this new technology, the mRNA technology, to develop that flu vaccine. However, when COVID-19 hit, they said, okay, well, let's hijack this process. Let's use this same technology, but instead of us using influenza as a target, we're going to use SARS-CoV-2 as a target.
So basically they've been researching, they've been funding for years, right? And then when this came, they just tweaked it a little bit.
Yes, they tweaked it and they said, okay, well, can we use it for this? And let's see how we can use it potentially for this. So that was one of the ways in which they were able to speed things up.
Okay.
Another way in which they were able to fast track it, they absorbed the cost. So they said, we're going to go ahead full steam ahead. We're going to do phase 1, phase 2, and phase 3. And we're going to go ahead and sort out how we're going to produce it and produce it in these quantities, regardless of whether we know whether it's going to obtain FDA approval or not.
Because usually the reason why it takes so long is because they wouldn't do that.
Right. You would wait until, okay, let's do phase 1. Let's do phase 2. And again, keep in mind that phase 1 can take 2 years. Phase 2 can take 2 or more years. And then there's still phase 3, which I haven't talked about. So let me touch a little bit on phase 3. Phase 3 is the final stage of development before a company requests essentially product licensing. And it takes usually 3 or 4 years to complete. So during phase 3, you continue to conduct these studies in the phase of development, and you're now recruiting thousands of study participants, right? Because you're trying to broaden the population of people that could potentially receive the vaccine. So remember, in phase 1, it could be less than 100. Phase 2, you're talking about hundreds of people. Phase 3, now you're talking about thousands of people.
Okay.
And you want to see, okay, looking at and then the number of participants is calculated so that you will have actual statistical differences between the placebo-controlled group and the group that's going to actually receive the vaccine. And so these calculations, again, they vary. It depends on the frequency of the disease in the population. There's several different models that you can use to establish or determine what that statistical difference should actually be. So what was nice about both of these mRNA vaccines that were developed, they were tested in populations. So with the Pfizer and BioNTech, I want to say that there were 40,000 participants in those clinical trials. And then with the Moderna vaccine, there were about 30,000 individuals in those clinical trials.
Right.
And what was nice also about those clinical trials is that there was a good representation of Black and Brown people. So in terms of Native American or indigenous people, those of Latinx or Black or in other African or Afrocentric groups, um, they made up about 40% of the population in which these vaccines were tested. So
Wow. Okay.
that was so for all of those groups. And I would say if you wanted to parcel that out and just look in terms of how many African Americans or how many Black individuals were included in the group, I would say it was 10% for Moderna and 10% for the Pfizer BioNTech mRNA vaccine.
And usually clinical trial part 3 or the stage 3 takes long because it takes long for people to
Yes.
volunteer, right?
Yes.
But now
Recruitment. Yes.
but since so many people were like out,
Yes.
so much recruitment happened, that's
Yeah.
another reason why it happened so much faster than normal.
Yes.
So what you're telling me is that you trust the process. The process had been going on earlier,
Yes.
and the circumstances that we were all in, specifically in this time, allowed for us to get those parameters done in a quicker period of time.
Yes.
So
Yes.
no shortcuts.
No
And
shortcuts.
I'm not
In
saying we're
fact,
God or anything. I'm not saying I
no,
trust
no.
them to the point where I trust God, but you have to get to a point where you do trust the process. And what you were saying earlier about the MMR, it's funny. How many of us have the measles, mumps, rubella shot? How many of us, when we step on a nail, we quickly run to go get the tetanus shot, right?
Right.
We're so trusting of other types of vaccines. It's just that this is so new, or we feel it's so new that we are
But there's a lot of, and another thing. So, I mean, again, we're going to have to see the long-term effects. They're going to continue. There's going to be a phase 4 of the clinical trials to establish what those long-term safety and efficacy effects are, or adverse reactions, whatnot. That will continue to be monitored in the population. One thing that I felt very comfortable about, and I would go so far as to say that if it was made available to me, I would take one of these mRNA vaccines. That's how strongly I feel about the data. The data is very strong, and there's a lot of safety and efficacy data. Another thing I wanted to mention was the amount of safety and efficacy testing that was done with these particular virus vaccines is actually a little bit more extensive than what we've seen for vaccines in the past.
Okay.
So usually you will look, if there's going to be an adverse effect or a strong an adverse event, usually that will usually surface or present itself within 2 months after the last dose has been administered. And so they were able to, in these clinical trials, both the ones conducted by Moderna and the ones that were conducted by Pfizer BioNTech, they carried the studies out and they evaluated and looked to see, okay, were there any adverse effects within this time period that are troubling, disconcerting, that would give us cause to want to pull this vaccine or continue forward. And the data looks very, very strong. So yeah, I don't have reservations. And when I actually got a chance to view the data that is publicly available, it looks very, very strong.
Yeah, so just to let you know, on Phenomenal STEMist, feel free to like and subscribe. Phenomenal STEMist on Facebook. I actually posted both, at least the approved ones. So the 2 mRNA vaccines, Moderna and Pfizer, have been publicly available by FDA.gov. If you want to know if you want to look at it yourself, it's the same things that the physicians actually read to see the adverse effects, when the adverse effects happen, etc. You can go to Phenomenal STEMist on Facebook. The 2 that have not been approved as yet, they are in stage 3, right? So Akuna was able to, Dr. Hanasho was able to explain what stage 3 is. That's the Johnson & Johnson and the AstraZeneca one. You're not going to have that information as yet. Oh my goodness. Everybody give some loves and likes for Dr. Hanasho, Dr. Akuna Hanasho. Listen, I have a couple of questions for you. If you have any questions, guys, feel free to ask. We have just a little bit of time for questions. The first question is there a difference in validity between established, approved, and then the ones that are pending? Like, I guess like which ones are better?
Well, no, see, they will all have to undergo approval. So I mean, the approval process is as rigorous for one as it is for the other. What I liked about the mRNA vaccines, another thing that I liked about them that also helped to fast track them, was the fact that they themselves are the vehicle. So with a lot of other vaccines, you're developing essentially a viral protein, and you're using some other host system to produce this viral protein. And the host system that you're using, it might view this protein as toxic and it doesn't want to make it. So there's a lot of hurdles involved in the development of these proteins. So what was nice about the mRNA vaccines, you're kind of bypassing that process because you're not asking, you're not,
you're not creating a vehicle to
yes,
put the mRNA in.
correct.
So
it
that
is itself the vehicle.
Yeah.
Yes.
That's the key thing. And that's why if I was to get a vaccine, I would get either Moderna
I was
or
gonna say, If
Yeah.
I want the mRNA ones, I don't want the other ones. If I'm gonna be honest,
the other one is a virus.
Not that there's anything wrong with them, and I'm gonna say, like it has to go through. So the safety and efficacy, it still has to be performed. It, regardless of what technique or what system is used to create the vaccine, it still has to go through rigorous testing. So in terms of the validity, I haven't seen the data package. Again, it's still undergoing those trials. It's not here yet. But I wouldn't be afraid of it. The safety and the efficacy, it has to be there, or the FDA or the other regulatory authorities are not going to approve it.
And I believe the viral vector got approved by UK's regulatory authority, I think, last week. But it's not here yet. But to your point, neither of them are robots, guys. So I know that's one of the constant, like, oh, they're gonna put something in us, like especially the mRNA. It's literally a lipid covering, like it just set it in.
It's like you put literally they put some fat around it, some lipids, some fat around it, and that's how it's getting in. So
that's all it is.
That's the beauty, I think, also of that particular type of method of vaccine development. That is the beauty of it. So if I had it in my power to choose, I would want one of the mRNA vaccines.
Adverse effects.
That's another thing I wanted to touch on. Not necessarily adverse effects, but talking on just the I think a lot of people, once they get a vaccine, they think, oh, I'm not gonna get sick, or I shouldn't get a fever, or there shouldn't be any response. When you get a vaccine, if you have chills, if you have fever, if you have muscle aches, if you have a runny nose, that's good. That means that your immune system is working and it's doing what it's supposed to do.
Yeah.
That's another thing I want to try to communicate, because I think a lot of people think, well, no, if I'm getting the vaccine, then I shouldn't experience any of these symptoms. No, this means that your body is priming, and it's working, and it's functioning, and it's doing what it's supposed to do, so that it can protect you against this.
That's a good ending. That's a good. It's doing. You're triggering an immune response. Everybody asked me this last time. A couple of people asked me, what are ways that I can activate my immune system? And I would say immunization. Like everybody's trying to do, the natural way. Of course, the vitamin C, the vitamin D, the vitamin whatever, and they're not connecting that the vaccine is it's active. That's what it's doing, guys. It's not. They're not just putting something in you, just to be mean, right? They literally, the very thing that you want to happen, which is to activate your immune system, that is the mindset of what the vaccine is doing. And so when you get those effects, it's really, it's supposed to be activating your immune system. So the flu shot gave me the flu is normal.
That's a whole other discussion. Because with the flu vaccine, I mean, it's a cocktail, and it's a hit or miss. One of the differences to, like, the coronavirus is different. So SARS-CoV-2, COVID-19 is the disease resulting from infection by a coronavirus. Influenza is caused by an influenza virus. So the coronavirus, it functions differently than the flu virus does. It belongs to, that's a whole other separate discussion, and then we're trying to close up. So let me just say they're different. With the flu vaccine, I think there's still more work that needs to be done. The flu vaccine, so, and just to give you an instance, so for the FDA to consider that the flu vaccine was effective, typically it only has to have 50% efficacy, whereas this virus has 95%.
95.
So just to give you a snapshot of how, if you would typically take the flu vaccine, that one, it just had 50% efficacy. This other virus, this other vaccine, I keep saying that, this other vaccine has 95%. Which one would you
choose? It's more fundamental. But yeah, I think her point was they get a response. They get a immune response thanks to
Yes,
the vaccine. And all we have to say is, well, like, that means your immune system is doing a good
it's
job.
doing what it's supposed to do.
Your
Yeah,
immune system is doing its job.
right.
Oh my goodness. Thank you so much, Akuna. I really appreciate this conversation.
You're welcome.
Once again, guys, feel free to share, like. I had, a good amount of questions, good amount of audience here. So I'm glad you guys were watching. I'm glad. I hope we were able to clarify vaccine development, and not just vaccine development, but like Joni said, the humanness of Akuna. She is a scientist. She is a woman. She is a Black woman. Just like me and you. I am the phenomenal STEMist, and so is she. So, good night, everybody.
Good night.